So again, welcome everyone. Here's Nadine, and I'm happy to welcome you to our webinar today, how to sensitively detect your target DNA for GMO analysis and food and feed matrices. So just before we start, a couple of maintenance, let's put it that way. So if you have any questions, please use the question box in your GoToWebinar panel, where you can just type your questions, and we'll definitely take some time afterwards to respond to all of them. If we don't have time, please don't hesitate and send us an email to kids@eurofins.com, and we'll make sure to catch up with you on your specific requests. So, and before starting, actually, I wanted to introduce the two of us with you, whom you're talking to. So my name is Nadine. I'm leading the team of the product managers here at the Eurofins GeneScan technologies team in Southern Freiburg. And I'm together today with Anja, who is the expert of the team on GMO analysis. And I'm happy to share the session with her, whilst I'm gonna do a bit of a start and then handing over to her to give you a bit more insights on our testing strategies. So, but before doing so, who are we, to be honest, besides the two of us? So we are Eurofins Technologies, providing of diagnostic test kits, lab consumables, industrial leading ELISA-based instrument platforms for the applications, as you can see, industrial solutions, animal health, as well as clinical diagnostics. So we are part of the industrial solutions teams, and I'd say the basis and the DNA of the team over here is definitely the GMO analysis, and that's why, hence why we're here today, and happy to present to you our solutions in the next couple of 30 minutes, and then we'll definitely leave enough time for some questions and some answers from you. So, short agenda of today's talk. So I'll give a short introduction on why we are here, what's GMO, and why are we testing. Then handing over or going a bit more into the automated DNA isolation, a couple of solutions we do have, as well as some technical insights, then handing over for the real-time PCR solutions for the GMO analysis before we're wrapping up at the end. So as an introduction, so who are our customers? I'd say that's the main question. And when we think of starting with a seed company on the left-hand side, because seed companies are prone and driven by creating new seeds that will definitely resist all these climate changes that are out there in the world, hence why developing more and more and different types of seeds that they mainly potentially want to export and to sell. Although within the world and the global world, we do have different regulations on GMO and GMO seeds, hence why they're often closely connected to service labs and testing labs that do follow then the legal limits of GMO contents in seeds. But the seed company is definitely also selling their seeds to the farmers that are happily growing the seeds on their farms. And then they also have an interest either in exporting those ones, but hence also need to follow export and global regulations in terms of GMO content, but also will sell parts of them to manufacturers, being it feed manufacturers that will potentially sell their or will rely on different, let's say, legal regulations, potentially allowing a higher percentage of GMO versus food manufacturers, where we do see stricter regulations. And both are interested also to export as well as testing to comply with global legal GMO standards. And then finally, everyone is selling into the retailers, where we as consumers are consuming foods of different natures. Hence why it's not only, let's say, legal status that needs to be complied with, but also food safety aspects, because we as consumers also do expect certain levels in safety. So this is a bit of the food chain, as we said, where we do see, okay, we as suppliers of technical solutions in order to detect GMOs, we interact with all of them at the different levels, especially in the testing laboratories, where we want to present and give the solutions to sensitively detect the GMO in the food samples. Now comes the questions how we can support. So we can support in terms of the different solutions as depicted over here. As I've mentioned earlier on the earlier slide, the matrices can be various. So it can be seed coming from a seed company, but it can also be a corn from the farmer or an end product that goes into retail. Hence why all these different sample matrices are then influencing the subsequent steps, as well as in parallel that the solutions we are offering, they need to comply with, and we do need to offer solutions that actually adapt and are validated throughout the process with these different matrices. As a next and critical step is definitely the sample preparation and the grinding, because it's important to grind your sample before it goes into the lysis and before it goes into a DNA isolation step, to have it in a format where you can then subsequently go on with your DNA isolation, which is a critical and often I'd say neglected step, because you need to sensitively isolate your DNA in a status that then you can follow up with your subsequent analysis, such as real-time PCR analysis, as well as having specific tools for your final data analysis, which is important to have in order to take the right conclusions on your presence or absence of your GMO. Having said so, let's go a bit more into details into the DNA isolation as well then into the real-time PCR offerings. As I said, a bit of a neglected step, but definitely a challenging step in this overall process. So what we do see when customers are coming back is what are the challenges they need to overcome. So there's this high diversity of matrix types as shown beforehand. So it can be starting from a seed, feed, food, different matrices, up to really highly processed matrix samples. And then hence why there are a high number of workflows already in place, really adapted to these different matrix types. And also labs coming back to us saying the training of the new staff is quite challenging because of this high variety of workflows, because of this high diversity of different matrix types. So we're driven by finding the solution and building the bridge in this case to give the support to you by harmonizing the workflows, by facilitating the workflows that are meeting these needs and this high diversity of matrix types. And let's have a look how we do it. So why do we need it, first of all? So we need a profound DNA isolation because this results in sufficient DNA that we need in order to meet real-time PCR sensitivity. And why that? Because imagine we want to detect the one GMO corn in your 10,000 non-GMO corns. So we really want to be that specific that we are able to detect this one in your sample preparations. So we want to meet the sensitivity of 0.01%. The DNA we want to analyze also needs to be intact, because if you have chopped short repeats DNA, a subsequent real-time PCR analysis won't work. So hence why we're really targeting at fragment length of at least 100 to 150 base pairs that we need to meet with a sufficient DNA isolation method. Furthermore, the DNA needs to be clean, because imagine with all these different matrices, there are also a lot of inhibitors that do come with that process that we do need to get rid of, because they will influence also the subsequent real-time PCR step. How we do it? Differently with different chemistry, but mainly also using extensive washings with dedicated washing buffers, and also providing a potential use of post-cleaning columns that can be used on the DNA eluate in order to clean it in case some inhibitors are still present. So how do we do it? Now we know why, but how do we do it? So it's nothing new, let's put it that way. Really open, because that has been invented and there are patents published back in the 90s by capturing in DNA using a surface that is silica coated. So negatively charged DNA is bound to a negatively charged silica coated surface. How that? So our solutions differs a bit. So we are using, instead of a silica coated surface, a carboxylated but still negatively charged surface bound to magnetic beads, because these magnetic beads will allow an automation. Because what we also want is to facilitate, not only to harmonize, but giving the customer the possibility to run it on an automation platform. So in this presence of an isopropanol, that helps to dehydrate the DNA, then facilitating this interaction of the DNA which is negatively charged in the phosphate backbone, with the negatively charged surface of these paramagnetic beads by sodium ions, which are building the bridge between these two negatively charges. So once we have the binding, we'll capture the DNA on these magnetic beads with magnetic rods, as I've mentioned, on an automated platform. Could be Kingfisher or an Autopure system, as we've been validating the workflow with. So hence these magnetic rods will then capture the magnetic beads where the DNA is bound, and then by implementing different washing steps, the DNA bound to the magnetic beads will be changed to different plates with different amounts of different washing buffers, until at the end they're going to get eluted into a elution buffer system. As easy as that, and it will take you in the lab, this automation step, less than an hour. So really hands-off time, giving you the possibility to do something else. So here's a bit the data I wanted to present to you today in order to convince you that we're trying to harmonize with a broad variety of matrices the workflows. And this is just an example of the workflows as we are suggesting, depending on the matrix type. So we really want to use it a bit more in a flexible way to really accommodate your needs. So again, let's start with a sample matrix. And I've picked two examples. So raw matrices, so could be a corn, could be rapeseed, as well as some intermediate matrices that are higher processed matrices. So first, as I've mentioned, it's important to use the grinding step. And here we're using a knife mill. So different solutions are out there on the market. It can also be down to an easy Thermomix you may want to use in your kitchen, but also in your lab to grind actually your food matrices. In the next step, there comes the lysis, which is also important. So here we're using a chemical enzymatic heat lysis treatment for approximately 1 hour with some spin down steps to really lyse the material and to release the DNA. So in some cases, and we do give some suggestion in which cases, we're adding a chemical to really put a bit more emphasis on those one that do inhibit the subsequent analysis, and therefore we do suggest a dilution buffer addition that is part of the offering. Before it then goes into the automated DNA isolation step, as I said, using an open automated platform by using these magnetic bead binding and respective washing of the DNA until we elute it in a defined elution buffer. In some cases where we do see a high potential inhibition because of the matrix type, we furthermore actually suggest to use a cleaning column just to get rid of some potential inhibitors before we determine the DNA concentration and the DNA purity by using a real-time PCR system. And the purity, we're looking at an internal positive control, which is in case of an inhibition shifted. And this is the indication we do analyze the data and say, okay, there is an inhibition shift versus there is a clean, pure DNA that can go into these subsequent analysis. And on the next two slides, I'm going to show you a couple of examples and outcomes of these analyses that, as I mentioned, hope will convince you that this is a solution that can be implemented into your lab. So what we've been doing. So in these cases, we used a 1 gram test portion in 7 milliliter of lysis buffer, and we subsequently purified the DNA out of 500 microliter of that lysate, coming out of what I was telling, a chemical heat lysis procedure. You see at the left-hand side the different matrices that were used: corn kernels, canola seeds, rice, soybeans, with the workflow as presented. So meaning, except using the chemical precipitation, except for the soybean, where we highly recommend using an additional dilution buffer because of the inhibitory matrix type. In these cases, we do not recommend to use any post-cleaning because the extensive washing with the dedicated washing buffers will release all these inhibitors in order to do the subsequent analysis. In the next column, you see the theoretical DNA concentration when we're really targeting the 0.01% of the sensitivity. Remember, the one corn we want to detect in the 10,000 of corns out there, that's what we want to meet. So it's theoretical based on the genome weight of the matrix type. In all cases, by using the workflow, we've achieved these concentrations. And when comparing to a workflow that is already in place in a routine laboratory using a similar system based on magnetic beads, also on an automated platform, we could even exceed the concentration, or at least meeting the performance of the competitor kit. So as a summary, we can say that the solution we are providing achieves the yield and meets the sensitivity when measured, as I was saying, with a specific real-time PCR. And as I've also mentioned, we were looking at the potential of the presence of any inhibition, and this was measured by measuring a CQ shift with using and looking at an internal positive control, which is included in the real-time PCR kits in our site. So this solution is called Imago Food, and this has also been tested on intermediate matrix types. So same setup. In this case, although we've used different input portions. So besides the 1 gram, we've also used the 2 gram test portion, which they're then diluted in 7 or hence 10 milliliters of lysis buffer. Again, you can see the same layout and same test design as presented beforehand. So we have different intermediate matrices that are higher processed than the raws, so corn flour with grits, expeller, pressed cake, rice flour, soybean meal, that went through the whole workflow, where you see also that mainly the core workflow was used, also with the addition of this additional dilution buffer. Now you see that in some cases we recommend, so in optional cases, again with the soybean, we recommend using a post-spin or post-clean column. In the case of soybean protein, it's definitely mandatory because we do see this high inhibition in these cases that we've indicated with the shift of a CQ value, but by using the post-clean columns, this shift can be eliminated. As well as we've been achieving all the DNA yield again, we wanted to achieve with either using the 1 gram in most of the cases or a 2 gram test portion. Again, comparing this case to a manual workflow, which is in place in a routine laboratory. And here you can also see that by using this automation and by bringing this workflow onto an automation platform, that will definitely also help facilitating the lab not only to run this manual workflow, but to exchange it to an automated. And in most of the cases, the DNA yield was exceeded or at least met the performance criteria as benchmarked here. I've also indicated what's coming next. So we're heavily working on completing and working on extending with different matrix types. So we are targeting food feeding stuff. We already have validations for food feed components, but we also want to get the whole food feeding matrix types. We are working on meat matrices, and then finally closing with highly processed matrices. So watch out, there are going to be some more application notes coming out in the next couple of weeks and months in order to get the DNA yield and the purity you need in order to run a subsequent real-time PCR analysis for your GMO in your food and feed matrices. So having said so, I'm happy to hand over because now Anja is going to present to you how does it continue the workflow. Now you have your DNA and what do you do with it?
So, okay, thank you, Nadine, for introducing the DNA extraction method. I would like to focus today on the GMO testing strategy and how you can improve your lab or your testing strategy so that you get really high efficiency. So we also have challenges for the GMO testing laboratories. First, we still have the high diversity of matrix types. We have high or low DNA content samples or samples that have a high or low GMO content. And for these sample types, the strategies are different. Also, for GMOs, the planting still increases worldwide. And in addition to this increase, we still have new approvals of GMOs with new modifications. And the GMO testing laboratory have to constantly adjust their analytical approaches and to have developed new assays. Also, the time pressure on food and feed producers affects laboratories. They have to permanently improve the efficiency of their lab. So how can we overcome these challenges? So first, before you start designing a strategy or even start testing your sample, you should try to get as much information as possible. So for example, is it very important to know the country of origin, because the global planting situation affects the GMO testing strategy. Also, accidental releases of GMOs, of non-approved GMOs in the country of origin has an impact on the strategy. The country of destination is also important because there is a legal framework and you have to know the legal situation in this country. There may be special analytical requirements, for example, non-GMO labeling that are required by non-GMO labeling organizations. The sample itself is very important. You may have samples with a high DNA content or very low DNA content. And as I said, there may be samples with a high DNA or GMO content. And there are samples that have botanical impurities. So for example, if you have a corn starch sample and there is a contamination with soybean, this may affect your strategy. And the most important thing is your customer, because they may have special requirements, and it's always good to talk with your customer first before you start the testing. So how do you find the most efficient strategy for your customer? I prepared several examples for you today, and I would like to show you the best analytical approach for different sample types. In the first 3 examples, I would like to show you a complete strategy, and this is based on the DNA content and the GMO content. So we'll have a look at example where we have a high or medium DNA content and a low GMO content, or a sample with a low DNA content, and how we can approach the GMO testing strategy. And then the second part, I would like to show you the first step in the strategy, and this depending on the sample type. So we will have a look at mixed samples and pure samples. So when you start to design a strategy, the achieve should always be to end the GMO testing at the very early stage, or the very ideally at the first step of the analytical strategy, and with as few assays as possible. So the first example, that's what we call the basic approach. That's an example where we have a sample with a DNA content that is medium or high, and a GMO content that is low, or we do not expect GM DNA at all. So in this case, it is best to start with a very broad screening and to cover as much GMOs as possible of this, the concerning species. And I will show you some examples in the following slides. If at this point the GM detection is negative, so have a negative screening result, normally the analysis stops at this point. And this is what you always should try to achieve for your customer. Stop the GMO testing strategy as an early point. So if it's negative, fine for you and your customer. If it's positive, then you could follow with an identification of GMO, use specific methods to detect, approve, or exclude non-approved GMOs. So if you, for example, detect a non-approved GMO, then you have to consult your customer or check the requirements in your country, the legal requirements, because sometimes there are special requirements that accept non-approved GMOs up to a certain threshold, but this depends on the country. If you have no non-approved GMO in your sample, then you may proceed with the quantification of the genetically modified DNA. And if the content is above a certain threshold, the product may have to be labeled. If it's below a certain threshold, then you don't have to label it. It depends on the legislation in the country. So this is an example where we expect no GMO content or a low GMO content, and it is highly likely that you don't have a positive result, and that at this point the analysis stops with this result negative. But what if we expect GMOs in the sample? Then the initial screening, the broad GMO screening, would to, with a high percentage, be positive. And this is no additional information for your customer. You have to choose another strategy for your customer if you have samples with a medium GMO content. So let's have a look at example 2. If you have a medium or high DNA content and a medium GMO content, it might be a good idea to start with the quantification so that you get in the first analytical step an information about the threshold or about the content of the GMO in the sample. You can start with the screening quantification and your screening targets or specific quantification. If you choose a specific quantification at this step, you should choose the most important GMO for this species. So for example, if you would like to quantify soybean, then it's good to start with a quantification of Roundup Ready 1 and or Roundup Ready 2 soybean. And I also will give you an example for this first level analysis in one of the following slides. If the content is above a certain threshold, then you should consult your customer and check the legal situation in your country. If it's below, then you can proceed with the identification to ideally exclude the non-approved GMOs. So the next example is different. We don't care about the GMO content because we have samples that have a low or no DNA content. So for example, soybean or cotton, soybean oil, lecithin, or cotton fibers. For this, it is very important to have an efficient DNA extraction method, because if your DNA extraction method does not extract as much DNA as possible, you will have negative results. And I will tell you what this means. So if you have a very low DNA sample, it might be a good idea not to start with GMO testing, but instead with the detection of the species DNA. So soy species in lecithin, cotton species in cotton fibers. If you have a negative result, and this is why you need efficient DNA extraction, if you have a negative result in some countries, maybe the requirement is that you have to prove by traceability that there is no GM corn or soybean involved in the whole product chain. And this can be rather complicated. So, but if you detect the species DNA and you have a positive soybean sample, then you can proceed with example 1 or 2. So you have, for example, soybean, then proceed with the screening, or if you expect GMO in the sample, then with the quantification. At this point of the presentation, I would like to mention the inhibition we could have. Nadine mentioned that in the DNA extraction. It can also, always occur that you have inhibition in your sample, that the PCR reaction is inhibited and you get negative results. So if you have a high DNA sample and you expect a GMO content and you have a negative result, the lab manager knows that he has to be very careful because it could be inhibited. But if you have a low DNA sample with a negative result, could come from an inhibition or because you have no DNA in the sample. So you have to exclude the inhibition. And our approach in this case is that we have an internal positive control in our assays. So if you have a result with a positive internal positive control and the negative species DNA or GMO result, then you know that it's really negative. If your internal positive control is negative, you should start to reanalyze or re-extract your sample. So if you want to design a GMO testing strategies, there are many ways, but you always should consider and know the current GMO situation and design the strategy in cooperation with your customer. The main elements of GMO strategies are screening assays. And if you do a smart selection of the targets, then it has a great impact on the efficiency of your strategy. So for example, if in the first example you have a broad coverage of the species, the GMO species, then the analysis starts at this first step. So you should carefully choose the targets for your screening assays. Let's have a look at soybean. So for soybean, 35S and NOS, that's the classical GMO testing screening, we have a coverage of a bit more than 45%, which is, I think, not good. You can add a third screening assay, that's the FMV promoter, but still you have almost 40% of commercialized GM soybean that are not covered by this strategy. We decided to replace the FMV promoter by our AgriBorder 2 element. That's a target that's in the border of a genetically modified construct. And with the replacement, we get an additional coverage and we achieve more than 80%. So still, there are some commercialized GMO soybeans left that are not covered. And for this, we added additional assays like the CSVMV modification and 2 event-specific detection methods. So for a GMO testing lab with this strategy needs 6 single assays to cover 100% of the GMO soybean. If you're fine with this, it's okay, but we think to enhance the efficiency in the lab, it's good to have less assays. And for this reason, we combined them in 2 multiplex assays. So we have 1 multiplex assay covering the screening targets and 1 multiplex assay covering the additional targets to achieve 100% of the commercialized soybean. Corn is a bit different. So if we have a look at corn, the classical screening 35S and NOS is really good. If we add FMV promoter or AgriBorder 2 as a target, it does not have an effect on the strategy. There are still 3 corn events left, and some customers still ask for the detection of these corn events. So that's why we combined the 3 events in an additional multiplex. So for our recommendation for corn is to use the main screening multiplex and the additional multiplex covering the 3 remaining GMOs to achieve 100% coverage of the commercialized corn events. So you may ask at this point, why do we recommend a triplex with 35S, NOS and AgriBorder 2 when you don't need AgriBorder 2 for corn? So 35S NOS duplex would be enough, and we offer this. However, I will show you in the next slide why the 35S NOS AgriBorder 2 could be an advantage for the lab. So until now, we were talking about pure sample types, but GMO testing laboratories frequently have mixed sample types with soybean and corn or other species. So let's have a look in the next example at the sample type where we have a mixed sample with soybean and corn. We have a DNA content that is medium or high and a GMO content that is low or we do not expect GMO. And I would like to focus on the first step, the screening approach. So if you start with this multiplex 35S NOS and AgriBorder 2 and you add the corn multiplex, you will have a 100% coverage of all commercialized corn. You only have to add 1 additional assay, that's the multiplex for soybean, to achieve 100% coverage for soybean. So with only 3 assays, you have 100% coverage for corn and soybean. So the next example, I would like to have a look at the GMO content again. So we expect GMOs in the sample, and we have a sample type that's pure corn samples, and we have a DNA content where we extract enough DNA to perform a GMO testing. So we learned in the previous slide that 35S and NOS is quite a good approach for corn, and you may choose because you expect GMOs in the sample, so the qualitative approach is not recommended, the screening approach. You may start with the quantitative assay, let's say the 35S promoter and or the NOS terminator. So, but please note that it's not that you should not add the 2 results because some GMOs have the 35S promoter and the NOS terminator. So you should have a look at the single results and do not add them. We have quantitative simplexes. You may use quantitative simplexes, but for labs that would like to achieve a higher efficiency also multiplex assays, quantitative multiplex assays are available. So now that's the 5th example. Now let's go to the last example, what I think is the most complicated example, because now we are looking at the mixed sample type, soybean and corn. We do not recommend screening targets in this case. Why? Because 35S and NOS elements occur in GMOs from corn and in soybean events. So if you do the quantification of 35S in NOS, you will also detect the soybean event. So the result you get, the detected amount of 35S is higher than the actual amount, because you quantify 35S for corn and the 35S from soy is added. So in this case, we recommend to start with quantitative specific quantifications. And as I said before in the previous example too, recommend to start with the most important GMOs of the corresponding species. So for soybean, we recommend to start with Roundup Ready 1 and or Roundup Ready 2. And for corn, also choose the most important corn variety for the quantification. Also for several approaches, we have multiplex assays that helps you to enhance the efficiency and to reduce the turnaround time. So in summary, how can you improve your GMO testing laboratory and avoid a lot of assays, to perform a lot of testings, and yes, reduce the turnaround time? So first, choose the best analytical approach, and depending on the expected DNA content and the GMO content, whether to start a qualitative screening or a specific identification. There are a lot of approaches. If you have questions, please contact us and we will help you with this question. Also take into consideration the species listed in the sample, whether it's soy or corn, both. Do we have a flax in the sample or rapeseed? Choose an efficient DNA extraction method, especially if we have samples with a very low DNA content or with samples that have a lot of inhibitors. Choose the efficient screening strategies with multiplexes. You don't have to perform 10 different single assays, just reduce them with multiplexes. And combine the results of the screening tests. So for example, if you have a GMO that is 35S and NOS positive, because it has both elements, and your screening result is 35S positive but NOS negative, then you know you don't have to identify this GMO and you can reduce the assays in the next analytical step. Use screening quantification for pure species sample types, and use specific quantification if you have mixed samples, so that you can avoid the additional quantification of the element in the other species. So, and with this, I would like to hand over to Nadine again for a short summary.
Thank you, Anja, and I hope we deciphered a bit the word of the GMO testing for you in the last couple of minutes. And I'm just wrapping up because started with that slide. I want to end with that slide showing you, okay, GMO analysis is not just the PCR, but it involves definitely upfront sample treatment, looking at your sample that is involved, the different, as Anja mentioned as well, then the subsequent DNA isolation, and then looking at the content of the DNA. So are you expecting a low? Is it a high process matrix or not? Can you expect a higher DNA concentration before you're entering then your DNA analysis using real-time PCR? So with the offerings we're having as shown here, so an automated solution as well as the different real-time PCR solutions, we can support you testing your GMOs. We did not talk about the data interpretation, so we definitely have also different tools to support you with that, because it is difficult. And as mentioned, we are happy to take now some questions and happy to support you in your GMO analysis. So thank you for your attention, and we are open for some questions.